In this video, we will discuss about cardiac arrest and the inflammatory cascade effect because of cardiac arrest. Let us start with this left figure. The cardiac arrest will lead to the oxygen tension, or we can say the deficiency of oxygen will be generated because of cardiac arrest, and this will lead to less ATP, less formation of ATP, and we know that ATP is the uh, main source of energy for the cells. So this will cause energy deficient condition for the cells, and cell will start to dysfunctional, such as cell membrane will not being able to perform its normal function, and the microvascular permeability increases, such as potassium starts to leak out of the cells, and there is exchange of water increases that will cause, and finally that will cause cytotoxic edema condition. And the cell functioning interstitum, that is cells that are present between the principal organs, or no, cell functioning interstitum, it means the tissues that are present between the principal cells of the organs starts to dysfunction, or we can say they will stop behaving as a normal cells. And another thing that will it will cause is that the oxygen exchange will decrease, so it will create a oxygen surplus condition at one point, and at the other end it will create oxygen deficient conditions. And the cell capillary distance reperfusion will increase. This is the condition, uh, this is the thing that will occur after the cardiac arrest, when the body needs to when body is restabilizing itself. So during this process, the blood flow between the vessels fluctuate, and this will cause cell capillary distance reperfusion. It is similar. This condition is similar when the similar to the this condition is similar to the reperfusion therapy. We will understand about this reperfusion therapy in some other video. And when the cell starts to recover, around 70 to 80% of the ATP production is recovered, then during this process the neuronal death will occur, and by the time it is recovering this ATP, the other conditions such as ROS, RNS will activate, will be high, as the because of the oxygen surplus condition present. ROS is the reactive oxygen species, and RNS is the reactive nitrogen species. And the cell death and increase in this both of this ROS and RNS will leads to activation of cell death signaling pathway. So during this period, cells are still still cells are still prone to death, or we can say programmed death, that is apoptosis, or uh, that is apoptosis. Uh, and another pathway that is activated, that is mainly describes about the functioning of the within the functioning within the cells, that is the calcium homeostasis will get disrupted, and this will leads to mitochondrial disruption. At the same time, we know it will also cause ER stress, because endoplasmic reticulum is also responsible for maintaining calcium homeostasis with the help of circa endoplasmic pathway. And this mitochondrial disruption will lead to uh, no production in the ATP, as mitochondrial is known as powerhouse of the cells, and its dysfunctional will leads to less energy deficient condition for the cells. The cytochrome C will also get released into the cytosol, and we know in our previous video from our previous videos that what are the problems that it might cause when the cytochrome C is releases into the cytosols, and ultimately all this will activate the cell death. And in some way, this intracellular pathway and the intracellular disturbances and the extracellular disturbances are connected to each other. as both of them will finally lead to activation of cell death. Let us understand the cardiac arrest effect on the brain. So when the cardiac arrest is been observed, the brain starts to release cytokines to take care of these disruptions or sudden changes. This thing happens within first 3 hours. So it will activate, increase the lactate and increase the signal interleukin 6 and TNF receptor 2. At the same time, the resident microglia that is continuously patrolling around the brain cells will get activated within minute, and it will take care of the macrophages that are being produced near the brain. And after days, it will start to and after days, if it will start to it will after this, even if the macrophages are not been completely repaired, then it might lead to secondary neural injury and neuronal death. And this secondary neural injury can also be caused by other factors such as increase in the cytokines and inflammation, and the cascade effect of this inflammation will leads to this secondary neural injury and neuronal death. And this inflammation will also cause ROS, that is reactive oxygen species. And because of inflammation, the immune system will get activated, and the excessive activation of the immune system, such as the excessive activation of T lymphocytes, will cause side effects that might also cause this secondary neural injury and neuronal death. And this neutrophil, during the cardiac arrest, this neutrophil got increased, and because of the activation of this immune system, because of inflammation, the neutrophils increase furthermore, and this will cause excessive increase in the this will cause uh, a negative effect on the cells, as the amount of neutrophils are increased beyond a safe limit within the near the brains, within or near the brains. I hope you have understood some of the changes that observed during cardiac arrest and after cardiac arrest, and the inflammatory cascade effect, especially on the brain. If you want to understand more in details, you can go through the reference that has been provided in the description. Thank you for your time. That's all for this video.